Good afternoon, everyone. I'd like to welcome you to this edition of the HNRCA Monday Seminar Series. I'm Dr. Roger Fielding, the Associate Center Director. It's my great pleasure to introduce today's seminar speaker, Dr. Johan Auwerx. Dr. Auwerx is a professor at the École Polytechnique Fédérale de Lausanne in Switzerland, where he holds the Nestle Chair in Energy Metabolism and directs the Laboratory for Integrated and Systems Physiology. He uses a systems approach to map the signaling networks that govern mitochondrial function to understand metabolism in health, aging, and disease. Much of his work focuses on understanding how diet, exercise, and hormones control metabolism through changing the expression of genes by altering their activity of transcription factors and their associated cofactors. His work was instrumental for the development of agonists of nuclear receptors, a particular class of transcription factors, into drugs which now are used to treat high blood pressure, lipids, fatty lipids, and diabetes. Dr. Auwerx received both his MD and Ph.D. in molecular endocrinology at Catholic Université in Leuven, Belgium. He was a postdoctoral research fellow in the departments of medicine and genetics at the University of Washington in Seattle. The title of his presentation today is Cross-Species Genetics to Map Players in Mitochondria and Aging. Johan, the floor is yours, and we're really looking forward to your lecture today. So thank you for joining us.
Thank you. Start sharing my screen here. Does that work? Okay, thank you very much, Dr. Fielding, for this eloquent introduction. And it's my pleasure to be here. I can make it given the circumstances, COVID, the war in Ukraine, but I'm still happy to give a lecture today. What I'm going to try to do is, instead of going on the latest, what has come out of the lab, I'm going to give you an historic overview of how we have been using actually genetics in the mouse and sometimes in the worm to elucidate new targets in mitochondria and aging. And about 12 years ago, when I arrived in Lausanne, it's about the last time we ever used a knockout mouse. So we started to start working with mouse genetic reference populations. And I'll illustrate this via the BXD genetic reference population, which I abbreviated GRP. So this population was created no more than 40 years ago by crossing a C57 black mother with a DBA father, and then the F2 offsprings were inbred for about 20 generations. And there's now about 185 of these mouse lines available. They are actually clones. You can generate more of BXD1 or more of BXD79, but they're genetically diverse. And here you see a little bit how this looks. Just the coat color varies in these mice. But these mice have varying phenotypes all over the place. And so they're a very nice way to study actually phenotypic variation and try to map genes. And a few of the characteristics are they have about 5 million sequence variants, which is as much about as in a human population, such as the Finns. They have about 4,700 genes with protein coding variants, 2,000 with predicted high impact variants. So it's as if you were studying 2,000 knockouts at a time. What's most important, though, is that they are extremely well phenotyped, and there's about 6,000 clinical phenotypes collected through history on these mouse lines. And including in these phenotypes are a number of molecular phenotypes which are available in 35 tissues. And all this is basically squared away. You can query it into databases, www.genetwork.org, which is run out of the University in Tennessee. And we have another database where we deposit our data called systemgenetic.org. And going a little bit into these phenotypes, I would like you to imagine them as we have here are 183 different strains in the y-axis. And here we have about 6,000 phenotypes, which are aligned by the different labs who generated them. And we like to think it's like the mouse variant of electronic medical records, because what you see is that there is a lot of white spots, missing data. For instance, we care a lot about cardiometabolic muscular phenotypes and care a little bit less about neurological phenotypes. So you see that it's a really good model to mimic precision medicine. But where it distinguishes itself from humans is by the fact that there's about 35 different tissues where we have deep molecular data going from RNA-seq to proteomics available. And certain of the metabolic tissues are very well covered. Like for instance, in the liver, we have data on chow and high-fat animals. We have data on males and females, young and old, and fasted and fed mice. So 8 data sets just on the liver alone. And this sets it very much apart from the humans. This BXD phenome containing the clinical and the molecular phenotypes has over 300 million data points. And we've pre-calculated this and made these data available on this website. And I'll explain a little bit to you how this works. So here I have 2 graphs. On the top, it's listed GWAS and PHWAS, where we align the phenome, which is about 6,000 different phenotypes. And here we align genetic markers, where we have about 3,000 aligned. These dots are all significant associations or correlations. The darker the dot, the more significant. And you can query this matrix in a forward genetic manner. For instance, you can say, I'm interested in body weight, which is a morphological characteristic, and you can query the genome, what are the genes which determine body weight, and you run a Manhattan plot, which is provided by systemsgenetics.org, and here you see that RPL26 would pop out as a highly significant candidate for body weight determination. What we think is more relevant, though, is we can do the opposite and run what we call PHWAS, phenome-wide association study, and know base ourselves on variants in RPL26. Why is this relevant? Many of you probably already have some genetic profiling done, 23andMe, Nebula Genomics, etc. So you know your genotype, and then you can query in this way. You can say, I have a variant in RPL26. What are the diseases which I'm likely to suffer from? And you see that we obviously also identify body weight as we do here, but we also have fat mass and VO2 now popping up. Now this is common practice, GWAS and PHWAS in human genetics. What's less common is transcript-wide association study or protein-wide association study and expression PHWAS. And the principle is, though, very similar. We have our 6,000 phenotypic traits in the y-axis in this matrix. And no, instead of having more genetic markers, for instance, we have here our transcriptome arranged to chromosomal localization. And again, you can query it in a forward manner. You can look, for instance, what determines VO2 max in my mouse, maximal oxidative capacity. And you see that SLC25A10 is a candidate for this in the liver transcriptome. You can conversely also say, what are the phenotypes where high expression of SLC25A10 is associated with? And again, you find VO2 max, but now you also find fat mass and body weight. So we advise every one of you to query this for your favorite gene of interest, because you might find really hypothesis generating data. And so we've been playing with this system in the BXDs now since over 12 years. I've carried out many studies and I don't have the time to go over all of them, but I would like to spend some time on what we've done in the BXDs. And we have metabolic phenotype them very intensively on chow and high-fat diet. And we've also looked at lifespan in the BXDs. And these are 2 studies which I would like to dwell some time on. So our studies are fairly simple in design. We enter cohorts at birth. Here in this case, we at 8 weeks put half of the mice on chow diet. The other mice received the high-fat diet. And then we align a number of phenotypic tests. We measure total respiration, we put them in the cold, or a glucose tolerance test, blood pressure, and we exercise, do a maximal exercise capacity at 23 weeks, where we, after that, we give the mice a voluntary exercise, we give them an activity wheel in the cage, and then we repeat this 2 weeks later, And at week 30, we sacrifice the mice and we generally collect 23 tissues. We've only analyzed about 4 of this study, so many tissues are still biobanked. And if you have a particular tissue which we didn't analyze, interested in, we're happy to provide you these tissues on a collaborative basis. So let me just go back one slide. This is how our data look. This is, for instance, an exercise where we looked at body weight gain on CHO in the yellow and high-fat diet in the red. And here you see the parents, the C57 black 6, and the DBA mice. And these are our different strains which we phenotype. And you see big variation. Some strains which don't gain weight, some strains which gain a lot of weight. One interesting feature is this is when we give the mice an exercise wheel. So you see most of the strains, but not all of the strains, exercise has an impact. And so we basically can map which are the strains or genetic determinants of exercise or will to run in these strains. And a more detailed look shows you a little bit the genetic variation which is happening. If you look at final body weight on CHO diet in yellow or high-fat diet, here we rank order the different mice, the different strains. You see immediately that this strain here is interesting because on a high-fat diet, it has a body weight which is perfectly normal as a CHO-fat mice. Whereas these strains here, for instance, you see that they have even on a CHO diet or clustering with a high-fat mice. And here you see 2 extremes, the BXD68, no body weight gain at all. This strain, BXD44, becomes as big as almost the rat. And here you see representative pictures. You need to imagine these are brothers and sisters. They have the same father. The only thing which is different is the way their genome is mixed. And you see, we think that this model, for instance, of obesity is a much more realistic model for human studies and more relevant and translatable than, for instance, studying a DBDB or an OBOB mouse. Now, one advantage of studying these populations is that they're permanent and we can expose them and control the environment very meticulously. And this is illustrated in this slide. I think if you retain one slide from my talk, it would be probably this slide, where we look at heritability and how environment impacts that. And here I calculated heritability of 8 different traits, and we will focus on white adipose tissue weight at sacrifice. And you see, when we look at males fed at CHO diet, we see an 11-fold range in white adipose tissue weight. Heritability is quite high, 0.65. If we now put the mice on a high-fat diet, we see that the range of variation of white adipose tissue weight decreases, but heritability increases because we stress the system and our genes are made to help us survive stressful situations. But what would happen if I would do the study in humans, like per 100 different people? We wouldn't control environment very carefully. We would be on different diets, have different exercise, and know, although the range of white adipose tissue weight is very similar to here, when we combine the animals of the high-fat and CHO-fat groups, we now only have a heritability of 0.2. And this you see graphically depicted here. You see a heritability of a 0.65 goes up on a high-fat diet, and then it plummets when we combine both. And now most of the variation is explained by diet and a little bit by environment interaction. And this means that in humans, if you would do the same study, you would not see this heritability. We call it invisible heritability because it's not missing heritability because you cannot see it. It's cluttered by environment. And I will give you a few examples of this during the talk. You see that many traits are like white adipose tissue trait, respiration rate, body weight, but other traits like, for instance, heart rate or body temperature or alkaline phosphatase levels, you see that here actually diet doesn't contribute almost nothing to the phenotypes. Now, this was also the first study where we started to apply multi-omics, multi-layer data analysis to the strains. And what we did in this case is we collaborated with Rudy Ebersol's lab in ETH Zurich, and we measured 192 genes at the mRNA and the protein level. And we did that both in chow diet, which are the lighter colors, and in high-fat diet. And you see the individual data displayed on the circus plot arranged according to the chromosomal localization or the mitochondrial here. And what I wanted to point out is a point about the heritability of expression levels of genes. If we look in the blue, which is the mRNA, and this is called eQTLs, the expression QTLs, we see that in the blue, in the middle part, you see that the expression of an individual gene is determined most in the cases by its own location, meaning it's in cis. The transcript is determined by the regulatory sequences. Now, if we look at the proteins, which are indicated in red, we see a number of them where they're predominantly determined by the genetic location, but we see much more traffic in the middle of this circus plot, meaning that protein regulation is more complicated, determined by transcription, translation, stability, degradation, which is impacted not only by genes, but also by post-translational modifications and many other things. So this was striking for us that actually protein regulation is much more complicated than gene expression regulation. We had here also a number of very interesting features, which you see here. These are QTLs, which are very high with a high lot score. And you see, for instance, this on chromosome 2 was a textbook example. We mapped on this locus a metabolite to aminoadipate. We mapped a protein level, DHTKD, and we mapped an RNA level, DHTKD1 mRNA. And we did this both in the light colors or in the dark colors in chow and high-fat diet. And this is, in fact, a monogenic trait where you see the B alleles. If you carry the B6 allele, you have low levels of this enzyme. The D alleles gives you high levels of this enzyme. What does this do? 2-aminoadipate is an upstream substrate from DHTKD in the lysine degradation pathway in the mitochondria, where it's degraded to acetyl-CoA. And if you plot, for instance, DHTKD expression versus 2-aminoadipate, we see a substrate enzyme interaction because low levels of DHTKD, you have more levels of substrate accumulating versus when you have high levels of DHTKD, no, the substrate disappears. But what was very interesting is that this 2-aminoadipate actually traced also with glucose. It was inversely correlated. High levels of 2-aminoadipate, animals with that had low fasting glucose. And if we dissociated our animals in groups which were normal or diabetic on chow or on high-fat diet, we saw that the diabetic animals always has lower 2-aminoadipate levels versus the normal animals. And this was done by HOMA-IR. So we could confirm this also in humans because we collaborated with the cohort Lausanne, which is a large genetic study in Lausanne, where they collected about 6,000 patients and monitored them longitudinally. If you look at the urine and determine 2-aminoadipate, we saw that diabetics had much lower levels. Now, this was by itself quite interesting, but it was not novel because about a year before we published our data, Larry Klisch from Harvard actually published a study where he showed that 2-aminoadipate was a biomarker and proposed that this was a biomarker of obesity. Now, why do we show this data? Because he did not have access. He did a study in humans and he didn't have access to deep tissues. So he couldn't look at liver expression of proteins and mRNA. And so he couldn't bring the 2-aminoadipate in connection with DHTKD. And this is the complementarity which I try to emphasize between mouse studies or model organism studies and human studies. So Rudy Aebersold was our collaborator on these studies, and in 2013, we could measure about 192 with SRM MOS-PAC proteins. In 2016, we applied actually SWOT-MS developed in Rudy's lab, and now we could measure about 2,600 genes at protein level and at mRNA level. And this is depicted here, and I will talk a little bit about the relationship with transcript and protein, because it's all given for us that transcript should correlate in protein levels, and nothing is less true than this basic concept of biology. Here we have 2,600 pairs where we measure transcript and protein. And you see that 1,600 of these, if we look at the correlation between the two, actually do not significantly correlate, the ones in gray. We have about 1,000 which are positively correlated, as you would expect, and about 200 which are actually negatively correlated. Since we have data not only in chow diet, but also in high-fat diet, we could plot these correlations in chow versus high-fat diet. And again, the majority here in gray did not correlate. There were a number which was positively correlated on both chow diet and high-fat diet, and a number which were negatively correlated. But the interesting ones were the proteins and genes which were actually negatively correlated or inversely correlated, where there was clearly evidence for gene by environment interaction. And I would like to draw your attention to this protein or this protein. Purine does not correlate at all on a high-fat diet, but it's one of the highly correlated proteins in chow diet. And you see this here. If you look at chow diet, purine transcript versus protein, you have a very nice correlation. This correlation is totally overridden by coming in with a high-fat diet. And CYP2B9 is actually exactly the opposite. So that means coming in with a high-fat diet can just override a genetic effect, which is evident if you would control diet. Now, one other thing which in Rudy's SWOT method was interesting to use was that he could cover about 67 of the electron transport chain proteins. And we all know the electron transport chain in the mitochondria that is composed of 5 complexes which convert energy provided in the food into and harvested in the form of ATP. But what is less well appreciated is that these complexes are not as we see in the textbook, but they are arranged as supercomplexes, where you have one subunit of complex 1 actually bonding with 2 complex 3 subunits and a variable amount of complex 4 subunits to make things simple. Now, there's one protein which is called COX7A2L, which has been posited that it was very important for the supercomplex formation by a group in Spain, the group of Tony Enriquez. And this has been highly criticized by the group of Niels Bohr and Larsen. Some said it is involved, some said not. And we were lucky that our DBA and B6 were divergent in the amount of supercomplexes. Here we run blue native page gels, so non-denaturing gels, and you see that the DBA mouse tray, you see, identify the complexes in here. This is what we call the supercomplexes, which are enumerated here. You see that C57 black 6 don't have these supercomplexes. And here you see representative strains, 32 or 39. This has supercomplexes. This has the B6 phenotype and has no supercomplexes. So postdocs in the lab, Poujaja, actually run 58 strains in these gels, and we could find a highly significant band, the QTL on chromosome 17 for these supercomplexes. Now, guess what was under this peak? If we looked at the protein level, we saw that COX7A2L exactly had a PQTL under this peak. And this is depicted by the reddish colors, where you see the liver and chow diet and high-fat diet have a significant QTL, a PQTL. And this is the strength of protein work, because if you were looking at mRNA, EQTLs, they did not reach the significance threshold. So showing you that measuring protein in addition to transcript is quite interesting. You would say, what's the big deal about this? Has this an impact? This has a very big impact, because if we look at in-gel activity of complex 4 and complex 1, you see that the strains without supercomplexes have most activity running as a 4 monomer or a 4 dimer, whereas these guys, which have supercomplexes, have much less of this, and they have their activity run as a supercomplex. And this traces again with the localization of COX7A2L. And so this shows you, by combining proteomics with these genetic reference population, we were able to solve a long-standing question in the field of mitochondrial biology. Now I'm going to shift gears a little bit and not talk about chow versus high-fat diet study. And now I'm going to focus a little bit on our work in aging. And another big advantage of working with these genetic reference populations, I said there are 6,000 phenotypes. So you can use the phenotypes collected by people who were doing a study 2 decades ago, the Hahn van Sant. The Hahn is still working in Groningen. They did a longevity study. And at that time, they saw a twofold difference in lifespan in about 25 of these BXDs, with a good heritability of 0.39. At this time, when they published their study, obviously the mouse and human genome were not done, and they could just show a QTL. But they didn't know what was underneath there. They didn't go any further. But now with our techniques, we could show that there were 70 known genes under this QTL. And we identify one mitochondrial ribosomal protein from the small subunit 5 as a candidate gene. And I'll tell you how we did that. That was Rico Houtkoper, a Dutch fellow in the lab, who managed this. So what he said is, if this gene has something to do with longevity, we would expect its expression to be changed during aging. And so he looked at muscle databases, and here you see MRPS5 has a nice inverse correlation with lifespan. If gene expression goes down by half, you live about 500 days longer. Second criteria which should be met with the longevity gene was that it should be working in different species. And so we knocked the gene out in a worm, and we had this. From all the 70 genes which we tested this way, there was only 2 which adhered to this criteria. One was a sodium potassium chloride channel, which is involved in the mechanism of action of diuretics, which obviously have a lifespan benefit, and this one. And you see that the lifespan benefit is quite high on this gene. What was furthermore interesting is this is part of a ribosomal complex. And although we had a genetic hit where MRPS5 was located, we saw that the entire mitochondrial ribosomal proteins, if we tested 12 of them, I just showed 3 here, behaved in a very similar way, both in expression or when we knocked them down. Now, we all know that 99.9% of all mitochondrial proteins are encoded in the nuclear DNA, translated in the cytosol and embedded here, and they take part of the 5 complexes of the electron transport chain. There is, however, this vestigial bacterial DNA present in the mitochondria, which actually gives rise to an mRNA, which is translated by these mitochondrial ribosomal proteins, which themselves come from the nucleus. And when these proteins are not there, you see that now there's a stoichiometric imbalance in the electron chain complexes, which is sensed by the cell as a stress. And the cell activates what's called the mitochondrial stress response, which is composed of the unfolded protein response of the mitochondria. And if sustained stress is there, we also see mitophagy happen. And so the question was, can we exploit these data in a pharmacological way? We can knock down MRPS5, and there's a study which we're collaborating with a Chinese group, which has a very interesting muscle phenotype, actually in the heart muscle. But we actually could take advantage of the bacteria within our mitochondria or bacteria within our cells, and there's a number of antibiotics which target bacterial translation. Doxycycline or the tetracyclines are a prototypical example. And if we treat C. elegans with doxycycline, we see that this mitochondrial encoded protein totally disappears, whereas a nuclear encoded protein does not seem to be affected. We see that this translates in a huge mitochondrial stress, which we can measure by GFP, and a significant lifespan benefit in the range of 35%. Now you say this is particular to the worm. No, this is not. If you treat a mouse primary hepatocyte or you treat a mouse with doxycycline, you see that here you also lose the expression of this mitochondrial encoded protein, where you barely affect the expression of a nuclear encoded protein, and you induce this mitochondrial stress response by inducing these chaperones. And we could even see across kingdoms, if we, for instance, take plants have mitochondria. If we water them with doxycycline, they have actually a lifespan phenotype. They live longer. And this is evidenced here by them retaining chlorophyll. much longer. No, there is one other way where you can induce this stress response, this mitochondrial stress response, and this happens in the opposite way. Doxycycline and MRPS5 knockout reduce mitochondrial proteins, but biogenesis, which we hear often enhanced with a compound called nicotinamide riboside that boosts NAD levels, does exactly the opposite. No, you have more mitochondria, and this results in a similar stress response. And here I use mouse primary hepatocytes treated with nicotinamide riboside. You see, no, the mitochondrial encoded protein is induced. No effect on SDH, which is nuclear encoded, but you see again chaperones going up. And this had a number of very nice beneficial effects. For instance, if we give a 700-day-old mouse, which is an old mouse, with nicotinamide riboside, if we supplement the diet with this, we see that they live longer. We see on a liver of a mouse in which we induce a fatty liver disease by feeding it a high-fat, high-sucrose diet. We see that we revert this phenotype. And even in the brain, if you look at neuronal stem cells stained with EDU, they are depicted here in green. They go down upon aging. Supplementing just aged mouse with nicotinamide riboside reinduces this. So how to explain this seemingly very different phenotype? So I showed you first that inhibition of the mitoribosome and inhibition of OXPHOS or natural aging lead to mitochondrial disruption. But we also showed you that nicotinamide riboside 31 activation, resveratrol or rapamycin activate mitochondria. They induce a mitochondrial, both these situations, activation and disruption, induces mitonuclear proteotoxic stress because there is a protein imbalance. And this will lead to the induction of UPRMT and mitophagy. And this, in a moderate level, will increase proteostasis and result in longevity. So with this data in the back of their head, Vincenzo Sorrentino in my lab, who's now working at Nestle, actually emitted the hypothesis, what if we would activate this mitochondrial stress, low-grade mitochondrial stress, by either treating mice or cells or worms with doxycycline or nicotinamide riboside to induce this unfolded protein response and mitophagy? Would we restore mitochondrial homeostasis? And the answer is obviously yes, because otherwise I wouldn't be telling you this. What Vincenzo did first was he looked in a worm model of Alzheimer's disease, and this is called the GMC 101 worm. It overexpresses the A-beta 142 protein in the muscle. And so when you treat these mice with nicotinamide riboside or with doxycycline, you see the induction of the unfolded protein response in the mitochondria and mitophagy. You see also that these mice which are treated with NR actually increase mobility. And that's the phenotype. If you overexpress the A-beta protein in the muscle and the worm, the phenotype is actually that they move less. And you see that on the level of the A-beta aggregates, there's a huge difference. Here you see the staining of A-beta aggregates and vehicle-treated worms versus NR-treated worms. There's almost a complete disappearance. So this is also happening in cellular system. Here we use the human SHSY5Y neuroblastoma cells, which overexpress in this case the Swedish mutation in the amyloid precursor protein. And you see in the control cells, you see these green dots, which is the A-beta, which is accumulating. When we treat with NR or with doxycycline, don't have time to show you the data, we see that these aggregates almost fully disappear. And even in the mouse, here we use the 2TG model with a mutation in APP and presenilin. Here we stain half mouse brains with thioflavin S, and you see the thioflavin stains aggregated proteins. You see a lot of them. If we treat the mouse with nicotinamide riboside, we see a very significant reduction in the number of plaques and number of thioflavin S responsive area. And this translates also into actually benefit on cognition. And we tested this with a test called the fear conditioning test. In the fear conditioning test, you give mice painful stimuli, in our case, an electric shock, in combination with a cue. In our case, the cue was either a sound or an air puff. And then you repeat this the next days, but in that time, you don't give them the painful stimulus, but just give them the cue. And then you see that the wild type mice will recall and make this link between the cue and the painful stimulus, and it freezes here in the red bar, a red curve, about 50% of its time. An Alzheimer mice doesn't remind this painful stimuli is associated with the cue, and he doesn't care less, and he moves. He is not freezing. However, if we treat that mice with nicotinamide riboside, and we have similar data with doxycycline, we actually recover this thing. So the question which Vincenzo and then Mario Romani posed themselves is, what happens? Is this a thing, this link between mitochondrial stress and aggregates? Is this something particular for the muscle or for the brain, or is this also in other tissues? We focused basically on the muscle, and the first thing they did was they collected the number of human data sets, gene expression data sets of human with aging, with muscular dystrophies or myositis and aggregation disease. And they saw always this inverse correlation with electron transport chain, respiration, mitochondria versus inclusion bodies, beta amyloid, chaperones, etc., which are always going the opposite. You find the same in the GTEx data where you look at females or males. These are supposedly normal people, not diseased people. You see again this inverse correlation and the same situation in the mouse. So Mario Romani started testing this. And what he did is he fed young and aged mouse with nicotinamide riboside, and then looked at A-beta aggregates with oligomer A11 antibody. And obviously, young mice, we were unable to detect antibody in the TA muscle. But in old mice, we detected these amyloid accumulations, and they were significantly reduced with nicotinamide riboside. And here you see the quantification. This was again associated with an improved mitochondrial function. Aged mice have lower mitochondrial proteins on chow diet. If we feed them with NR, we recover them and basically make them almost normal again. Now, these were mice, and he had also similar data sets in worms. But what I would like to show you is some data in humans, human myotubes. Here we show representative data of 3 myotubes from aged subjects, which we treated with nicotinamide riboside to increase NAD. And here we stain them with proteostat, which actually stains aggregated proteins, as you see here. And here in the blow-up, if you treat increased NAD levels, you lower these aggregates. And the same happens when you treat them with a PARP inhibitor, which also increases NAD levels. And even in a disease model, these are 3 patients with a disease called inclusion body myositis. In simple terms, it's similar to an Alzheimer's disease of the muscle with heavy accumulation of aggregates in cells here. And you see again, virtually normalization. So with that, I would like in the last 5 slides or the last 5 minutes, talk to you a little bit about some unpublished data which we generated on lifespan in another genetic reference population. So far, we've mainly been dealing with the Ph.D.s, and this is the intervention treatment program. Now, the ITP is actually a National Institute of Aging sponsored research program where investigators can have their favorite compound tested for impact of lifespan. It's actually a very well carried out study because it's done in 3 different locations, so excluding the effect of a single environment. And it's done in a genetically diverse F2 cross where it's generated, it's called the UM-HET3 mice, which is generated by crossing BAL and C57 like 6 to generate the mothers, and C3H and DBA to generate the father. So you mix 4 genomes. And in all these studies which have been carried out at NIH, there were a number of control samples there. That means mice which were not treated with the lifespan-inducing compound. And in addition, there were also a number of these studies where there was no effect of the compound of lifespan. And so we collected about 3,300 mice, 2,400 as real controls and 920 drug treated where there was no effect on lifespan. And in these mice, evidence data on lifespan, and then we had the data on body weight at 4 different time points. And the biggest effort, what we actually did in our lab, was the genotyping of these mice, because this is not an easy genotyping. You cannot put it on an array. You have to develop a genotyping protocol. The goal was to see, can we identify genetic loci which predispose to lifespan extension? And first, a few quality control studies. This is just the lifespan in females and males in this study. And you see that despite that our mice don't run with motorcycles or don't carry guns or do drugs, the males really have an early mortality, which is very different. The females actually stay healthy and then really catch up with mortality later on. And here you see the number of cumulative events. The other thing which was very interesting, where we were looking at the relationship between body weight and lifespan. This is a complicated graph. Here we have the body weight. Here the time when the mice dies, the lifespan. And on the top you have the females, and the bottom the males. These are all our mice which we analyzed. And actually we categorized the mice in different categories. The red, which die between 6 and 12 months of age, to the green, which die after 42 and 48 weeks of age. What's very striking is that you look at the mice which die early in the red or the orange colors, they all have a higher body weight than the mice which stay longer. In the middle of the life, body weight doesn't make a big difference, and it actually reverses the trend in older males. It's also present in females, still significant, but to a much lower extent. And the impact of these data becomes more clear when you look at the hazard ratio. Hazard ratio means the risk of dying for a mouse if it's 10 grams heavier. And in blue are the p-values for the males. You see a very significant, a 1.7-fold increased risk of dying when you're heavier, 10 grams heavier at 6 or 12 months. And this risk inverses. Now you have a negative risk here at this time. And this is, if you're heavy later in your life, your risk of dying is less. So we were puzzled by these data, and Maroon Bousleiman, who was executing on this study and developed the genotyping and did all the analysis, was wondering whether the same is true in humans. And now we resorted to a technique called Mendelian randomization. And we looked in the 500,000 plus subjects that are available in the UK Biobank. And what Mendelian randomization does is it analyzes the link between an exposure, in our case BMI or early body weight, and the outcome in the UK Biobank, it's parental longevity or BMI. We don't have longevity of the UK Biobank cohorts yet because they're too young. And you use to query this with genetic variants. And here you see the typical example to ascertain the link between LDL cholesterol and coronary artery disease. You can use genetic variants in the LDL receptor gene. And so if we look at the exposure, early body weight, which is a phenotype in the UK Biobank called body weight at 10 years, and the outcome is lifespan, we see that there is a negative relationship. So people which have a high body weight at 10 years, they live shorter. If the exposure is still body weight at 10 years, and no, it's BMI, the outcome, we see that people who are heavy at 10 years, they actually risk to be significantly heavier when they're adults. However, when we look at adult body weight and longevity, we see that there is very little impact on survival. So that means really the risk factor is early body weight gain and not body mass index at the end. And so this is the kind of studies where I like to try to show you and convince you that this combination of mouse and human genetics is really important. Now, there was one deceiving point in this study. Although we had 3,000 mice, over 3,000 mice, we did not map a single sex-independent longevity locus. The only one which matches was in 17, but we're not too excited for this is here. We had, though, female-specific and male-specific locus. A female-specific locus on chromosome 3 and 2 male-specific locus loci on chromosome 4 and 15. And Maroon is currently analyzing these to look at the candidate genes underneath there. So this is my last data slide. I hope that I convinced you that GRPs, the genetic reference populations, provide flexible gene and pathway discovery platforms, allowing the dissection of complex traits and gene by environment interactions that are impossible in humans. These experiments can be seamlessly integrated with human studies, creating this virtuous cycle between mouse experiments, human validation, human observation, mouse experiments, etc. I showed you also that 2-amino adipase is a biomarker for diabetes, DHTKD, a new candidate gene. COX7A2L is a genetic determinant of mitochondrial function with implications for cardiometabolic disease. And genetic or pharmacological activation of the mitochondrial stress response reduces proteotoxic stress and increases several health span and lifespan measures. And we also think that instead of single genes, it's more likely that processes, mitohormesis, proteostasis, inflammation, rather than individual genes, determine health and lifespan. And the last slide shows you the people who did the majority of the work. Broom Bousleiman did the IDP study, Hao Li, Mario Romani were in other recent work. And then we have longstanding benefits from two very brilliant fellows, Laurent Mouchirou, who introduced us to the worms, and Rickert Houtkoper, who made the observation on the mitochondrial ribosomal protein, and many other lab graduates. Christina Schonjans is my long-term collaborator and spouse, and we have been working for very successfully with Rudy Aebersold and Zoltan Kutalik, and this is funding sources and disclosures. I thank you for your attention, and I'm happy to take questions.
Thank you, Johan, for a beautiful presentation today. We have some questions coming in. I want to remind folks, if they have questions, if you can use the Q&A function, that would be helpful. Dr. Andy Greenberg, one of our colleagues here, highlights that this was a fascinating talk, and thank you. Please recall the amino adipase, any connections with that, or glucose metabolism and aging?
So we did not look at this correlation with aging, but I would say that high amino adipate levels probably would be protective for aging, because they would, I think, be linked causally with diabetes.
Yeah. Is active ingredient NAD plus or NADH, and why NAD plus rather than the reduced form? I guess this is in reference to the maybe the NR stuff. I'm not sure.
So I think the reason why we actually pay more attention on NAD than the NADH ratio is that the redox balance is clearly more important for enzyme activation. If you look at the different enzymes, NAD itself, we see it more important as a cofactor or co-substrate for enzymes. And I'm talking here about a certain family of enzymes, HDACs, or for instance, the PARPs or CD38. And that's where NAD definitely is more important, and NADH doesn't do that function.
And then finally, what proteolytic components are engaged, both autophagy, mitophagy, with or without ubiquitin proteostasis, and does that relationship change upon, I guess, the kind of stress or aging?
The relationship there is, we've been mainly focused on mitophagy, because we think that you have first this unfolded protein response, and then you activate mitophagy and autophagy, and I think that's beneficial. We haven't been paying attention to that much to the proteasome. In a few studies, we have looked at it, but very superficially. And I think it goes hand in hand. More proteasomal function actually is also protective. But we cannot say that firmly, because we did not do a whole lot of studies on this one.
Dr. Jiandong Wang says your cross-species validation study between chow versus high-fat diet is very interesting. Have you studied the aging component, such as young versus old in your study, which could help us to detect some candidate aging genes and pathways relevant to aging?
In the aging studies, two different aging studies which I showed you. I showed you one which is a reanalysis of the study of the Hannah von Salm. There was no diet effect in that one. And the ITP, there was also no diet effect. There we only had body mass. So we had body mass, but I cannot tell you about the diet they were eating. I can tell you though that in the ITP study, one interesting confounder was the mice which are heavy early on in their life. These are the mice which are probably deriving from a small litter. So they have more food access. Whereas the mice from a larger litter eat less. So, but this is very indirect. We don't have food intake. There is, however, one study where we go over, then there's a study by Rob Williams' lab, published last year in Nature Metabolism, where we looked at the high-fat diet. The high-fat diet obviously shortens lifespan. But this is not in any of the studies which I showed you here that we can make this direct link.
Question from Kristen James. Is the higher early mortality in male mice due in part to behaviors such as fighting, or do you believe it is mainly driven by biological factors such as the weight gain data you shared?
I think it's both. There's definitely a more tendency to be aggressive in male mice when you house them. So I think there's a number of traumatic deaths, also deaths in these mice. But I also think the body weight gain is important. And I just pointed out one thing there is litter size. So we were able in one of the sites, not at all the sites, the San Antonio site, the investigators looked at litter size, and litter size basically translates into body weight gain. So the smaller the litter, the more body weight gain, the more mortality. The larger litters actually survive better.
And Thomas Beater asks, how prominently were neuronal expressed genes associated with aging?
It's bad to say we did not look at the brains in any of our genetic studies. We collected brains, we collected whole brains, but we never looked at the link in the direct link where there is a correlation at neuronal gene expression and lifespan. We have, however, for certain of the studies, we have the brains biobank, but it's total brains.
So there's a question from Dr. Chris Wiley, again, really fascinating talk. And he's asking about, you know, the caveat about crossing different laboratory strains and that the alleles don't evolve together in nature. Have you made any observations using wild-derived strains, which would have both genetic diversity and conserved evolutionary interactions?
This is a very good question. It's exactly what we are. I'll share my screen again here. This will stop others. No, I can. Yeah, I cannot share my screen. So we are actually in the middle of a study where we have addressed this. So it's what we call the hybrid diversity panel, where we use some founder strains of the collaborative cross. The collaborative cross contains 3 wild type strains: WSB, PWK, and Castaneous. And so we're actually in the middle of a study which should come down in early 23, where we're looking at health span and lifespan also with these genes introgressed. The problem with these wild-derived strains is sometimes, and that's a really pervasive problem, they're from different subspecies of a mouse. So even if we introgress these genes, they're not very well retained. It's like, you know, because the subsequent mice are not fertile. It's a little bit the problem of the donkey, you know, and the mule. They cannot reproduce, so you lose these genes in the long term. But it's a very good point. We only have, these are mainly the studies we showed you so far, laboratory-derived strains. But we should have data in a year or so with some wild types genes.
That's really interesting. Dr. Wang asks, again, many cancer cells depend on NAD+ and SIRT1 to sustain their rapid growth. Is there any evidence that nicotinamide riboside might fuel cancer growth?
Good point again. in tissue culture, if you grow on an organoid, you add NAD or you add on a cell line which you have in culture NAD, they will actually proliferate more. So it sustains proliferation, but what in vivo, there's no evidence whatsoever, and actually counter evidence, NR or NAD supplementation reduces tumorigenesis. And this is because its main activities are in the niche or the immune involvement in the cancer. There's actually a very nice study from a group at CNEO in Spain where they looked at liver cancer, and it's actually protective against cancer just because the major predominant impact on immune function.
Interesting. Dr. Chao Kang Lai asks, have you conducted DNA methylation profiles for the GRP mice?
No, we haven't. It's on our list to do for the new study where we also looked at the wild type in the wild type sample. We haven't done in these ones. To look at the biological clock, we haven't done.
And Brian Bennett asks, the heritability data from the BXD mice indicates that diet is an important factor to consider. To extend across species analysis, do you think that genotype-guided interventions in humans can supplement larger cohorts like the UK Biobank?
So the question, I don't understand the question. So he wants, the person wants to see whether we have, whether diet data for the UK Biobank. Yes, you can look at those, but these are all questionnaires. So we don't control the environment. If there's any data in UK Biobank, it's basically data derived from questionnaires. So we cannot control it as much. The power of UK Biobank is the number. You see, so
I guess I think maybe this question, Johan's about the idea that are there genotypes that could guide sort of dietary interventions in humans, I think, like
yeah, definitely their genotype. And I think where I showed you like this correlation between transcript and protein, if you see how this can change, really go opposite, I would say, yes, there are genotypes where you could say, okay, if I were a food company, let's say Nestle, I would focus on these which are actually where you see the genetic effects overridden by diet, and there you would have a direct handle on something like this.
Respond differently to a high-fat diet, for example, based on their, exactly. Yes, thank you. Again, another question about the 3AA data suggests that dysregulation of kynurenine pathway may play a role in diabetes. Do you have any thoughts about
again,
this?
good point, because the 2-amino adipate is also part of the kynurenine pathway. And yes, this can play a role in diabetes because if you know very well, the kynurenine pathway underlies the de novo NAD biosynthesis pathway. So it's really tied with SIRT1 function. And we showed that previously in a paper in the kidney. Yes, I think this is also linked with diabetes.
And I think we have one final question, and I think this is a good sort of way to sort of think. I think this is from Dr. Mazepina, but how do you think generally about, I think you've shown really elegantly sort of working from the mouse to the human, but what are the things you're trying to be careful about when you're considering translating results from the mouse to the human? It looks like your approach is to look at a lot of these things in the mouse, but then try to find models or data available to try to understand them in the humans. But do you want to comment on that a little bit more?
No, this is a very important point. I think one of the things where should warn the audience about is using single mouse trains. So if you use a C57 Black 6 mouse, it's like studying the impact of an intervention on one single human. It's not more, it's not like a population. And that's why I'm saying there's very easy ways to do and look at this genetic diversity. For instance, the collaborative cross founders, these are just 8 strains. So if I see that my intervention, my knockout, does the same across these 8 different strains, I know it will translate to humans, and I know it's generalizable data. And this is where I think we're often too much focused on a single strain. Like for instance, you can almost not induce NASH in a C57 like 6 strain, but how many people have tested other strains? And that's where I think the field should be going. And believe me or not, this looks like big studies, but I've only done 6 studies and my mouse bill is lower than if I would do knockout studies, because it's like data which I can graft on data which are available from other labs which study the same strain. So by going into the BXDs, you all of a sudden get 6,000 phenotypes. Who can do 6,000 phenotypes in the B6 mouse? So introduce the genetic diversity. I think that's what's really important if you want to make the mouse survive as a tool for human biology.
Well, this has been a really nice discussion about utilizing these tools in metabolism, nutrition, and aging studies, and how to translate these effects. And I think they have important impact for a lot of people's work. So on behalf of the HNRCA, Johan, I do want to thank you for your really informative seminar and the discussion we were able to have virtually. So thanks very much. And thank you all of our attendees today. Our seminar will resume next Monday at noon with Dr. I don't know who the next speaker is. I don't know if we have a slide of that, but anyway, we'll see you all next Monday for the next seminar. You can check.
Okay, thank you very much. It was a pleasure to talk to you.
Okay,
Take care. See you and stay safe.
All of you as well. Bye-bye.
Bye.